Cornelia and the Tübingen team for organizing this webinar, and I just would like to welcome everyone to this session today, in which we will be showing the new interface of the ORDO Connect Genome-Phenome platform. As you know, this is a key infrastructure for the Solver-D project, and thanks to it, we have been enlarging quite a lot the data sets that are available in the system. I think that as of today, we are very, very close to 30,000 exomes and genomes in the system, if not more, with their corresponding clinical and phenotypic information. So this is not only a system that allows you to analyze the data, but also a system that allows you to share and discover data, and also to serve as a resource for the future and future research in rare disease genomics, especially regarding the research on diagnosis. Today we're going to be talking, we're going to be doing a webinar, a workshop, a tutorial to show you some of the features that are now available in the system and how the new interface looks like. Some of you might have already tested this new interface, and you will see it's very, very different from what we used to have. We hope that it facilitates the analysis, that it guides the user a bit more on what steps need to be done. And also we designed this interface to allow us to add other functionalities that otherwise were very difficult for us to add. So hopefully this also provides a new framework for us to add new functionalities in the upcoming weeks. I'll share very briefly the agenda here. So I'm just going to be giving you a very 2-minute introduction, which I'm doing now, about the webinar today. Then Leslie will be using the bulk of the time to present the new design of the platform. She will go through a case analysis, through a cohort analysis. She will briefly show also Matchmaker Exchange, how to generate a clinical report. And then also Steve, many of you know Steve also will be supporting her in this webinar, answering questions. So at any time, if you have questions in the course of the meeting, put them in the chat or raise your hand, and Steve will try to answer them through the chat. If he sees that he cannot do that through the chat, might stop the webinar to answer you. I think that the other thing I wanted to highlight is that this work has been possible, obviously, thanks to the work of the team at the FNAC, but also many of the Solver-D partners and members, which have been proposing functionalities, have been doing intensive testing of some of these interfaces. So this has taken us quite a bit of time to be able to release the production, because every time that you do new things, you discover things that can be improved, that are not completely working well. So thanks a lot to all of you that have been proposing features and have been helping us with debugging and identifying things that could be improved. One very important thing is that although we are doing today this webinar as part of Solver-D, a lot of or most of these developments on the new graphical user interface have actually been funded through EJPRD, the European Genome-Phenome on Rare Diseases. We will also be doing a webinar with them at some point in time, maybe in a couple of months. So I think that is also very, very relevant. And this means that all these developments are not available only for the ERNs, which are participating in Solver-D, but also for all the ERNs that are in Europe and that are those participating in EJPRD. I think that the people that have been mostly involved with this work has been Alberto and Davide, Tassos, also Dani on the part of the sysadmin, Marcos, and then on the side of feature proposal development testing from my team has been Leslie, Steve, Gemma, and Anida. But also, as I mentioned, many partners in EJPRD or Solver-D, also members of ERNs, which are maybe not directly associated with these projects, have also been helping us to debug some of this work. People from the Carlos III also very much involved in working with us on this topic. Also, I'd like to acknowledge, obviously, the ORDO Connect GPAP users in general, because many of you send us comments when you see things are not working or you want to see new features implemented. Also, the rare disease patients and associations for trusting us with their data for research, and Prioritise, who is providing the Aspera service, who is the service that many of you use to upload the data into the system, and this is something they provide free of charge for us to work on. So I think that with this, I'll finish this short introduction. Thank you to all of you, and I hope that you will like this new version, and that what Leslie will tell you will facilitate this entry to the new GPAP that we are presenting today. So thanks a lot, and Leslie, all yours.
Okay, thank you. So let me share. You need to unshare so I can share.
Yes, sharing.
I see in the chat someone saying that the audio is very bad. I don't know if it's for all or only. Well, let me know if there is any issue with the audio. Okay, so the idea of the webinar today is to show the new functionalities. So we won't be showing everything, but just the new things and where you can now find the information that we have redistributed or put elsewhere. So I will be doing the demo with the playground instance, so with the demo data. This is not the GPAP with all the cases you have in the system, but it's almost it's the same. So this is the landing page that you will have now. At the top, you will see the different type of analysis and approaches that you can do, and today we will briefly go through all of them. And here at the bottom, you will always have the different studies, the different analyses you have done that now can be saved in the system. So you will see that you can easily go back to one of your cases and continue the analysis exactly at the same point that you left it. So I will be dividing the demo in the different parts regarding the different analyses. We will be looking at the case analysis, so how to analyze a family. Then we will see the improvement at the cohort level, which is very similar to the search across all experiment, which if you have seen maybe in the platform is for now disabled because we have a lot of data and we are working in the server to make it available again, and it will be in the next month. But for now, you can still use the same application, just creating a smaller cohort of any type of individuals based on phenotypic or experiment data. Then we will show how to use Matchmaker Exchange directly from the GPAP. And finally, I won't enter into many details, but just for you to know that we have also a new module to create a clinical report directly from the platform. So for the case analysis for a family, you will first click here in case analysis. And as I mentioned, now you will be able to save the information in the system. So you can enter the title of your study. I will say case demo webinar. And now you have different ways or different IDs that you can enter to identify the index case you would like to analyze. Today we will be analyzing here the case 15C. So when I select my index case, you will see below that the system automatically queries based on the information you have entered in PhenoStore the other individuals from the family from whom we have data. So in this case, we have the father and the mother of this index case, and we can see that we have a trio then of genomes to analyze. Of course, you can decide if you want to analyze the whole trio, or if you want only to go from one individual or the other, you can delete the data. Then the next step, you will retrieve the current information that we have on this patient. So on the left, you have a summary of the phenotypic data that we have in the system with the HPO. So you can recall this case. You have the pedigree, and you will see also the information if any variant has been already tagged for this patient. You will see it here with the corresponding clinical significance. The next step, based on the information that you have from PhenoStore, is to select for the genomic analysis which type of inheritance you would like to assess. So what we have done here is to automatize the genotyping, so you don't have to click each time and make different queries for the genotypes. Now, for example, here if I want to assess an autosomal recessive inheritance with both homozygous or component heterozygous variants or a de novo approach, I can select both of them, and automatically you will see that the different genotype patterns will appear for each of the inheritance to assess. And you will be able to do all these queries at once. You don't have to redo it. And the next step and the final step will be to apply the filters. So the new functionality we have implemented here is that you can easily create preset filters, so you don't have to always go and enter all the information on the annotations you want to filter by. So here, for example, I can select one of my filter, which is here, first approach. And once I apply it, automatically I have pre-filled all the information, the variant type, the population, etc. And I don't have to go back there. And on the right, you have the information to create your in silico gen list. And here it's very similar to what you already had. So I will just go through the new things. At the disease level, as you know, we had PanelApp integrated. Now we have made it more user-friendly to browse across panels, meaning that you can click and see all the panels that are available from PanelApp. And then once you select one of the panels, you have the exact number of genes for the whole panel, but we also have information on the one that are in the green list and direct link to this panel in PanelApp in case you would like to have more information there. Then when I add the panel in the system, what the system does is take all the genes from PanelApp and it looks if these genes are compatible with the version of ensemble that we are using. And in this case, for example, you know that these two genes, the symbol is not compatible, so you will need to check for the version 37 of these symbols to ensure that you capture this information. Then we have also improved the HPO gen list functionality, meaning that now you can see the HPOs that have been entered for a specific patient. You can delete also any HPO that you think might not be useful or might give you noise in the gen list you are creating. And then once you have the HPO selected, you can create a gen list based on the different algorithms. This you had previously in the platform, the HPO ontology, or for example, DGNet. And when you select the HPO ontology, you can also intersect from each of the HPOs instead of having the list of all the genes associated to all the different HPOs. You can also intersect them to see if there is any gene, in this case it would be Tango2, that is common for all these symptoms. So this would be the main improvements at the gen list level. And now you can start the analysis. So this is the interface that we have now for the results section. So basically, based on the different inheritance pattern we have selected here, we have different tabs. So one would be the results for the autosomal recessive homozygous, meaning that the case is homozygous for the variant and the parents are carriers. We have the same for autosomal recessive component heterozygous or the de novo variants. If I click on another analysis, then I will go for the variants that are here, in this case, de novo. So if I go for the variants, I think I made a filter that I'm not getting the ones that I was expecting. Okay, so so here we have the data. You can then save your study here on the top left of the screen, and case demo webinar. So now I can save my changes and the study will be successfully saved. And this means that if I go back here at the home, then I can take one of my studies that I have launched and load the study. Now I'm loading a different one because I don't have the variants. I made another filter and I prefer to show you this one. But it's the same approach. So we have different inheritances and the number of variants. Here we have then in the results table, the SNV and indels, and then the CMVs. The CMVs are not yet available in RDConnect, but they will be very soon. So I won't really enter into details here. And I will be focusing on the SNV indels. I will select the de novo ones. And now here in the table, we have also included the information we thought would be more relevant for the user. So you can see it's a little bit different from the table you were having in the previous version. We have now the nucleotide and the protein change with the consequence. You will also be able to order the list of results. And you can also see all the transcripts associated to a specific variant by clicking in this arrow. So once you select a variant, you will also have all the information concerning the genotype, the depth, and the alternative allele frequency, the population information, the in silico predictor results, and information on previous tags. So in this case here, you can see a flag here, meaning that the variant has already been tagged. And if I click in the previous tags that you will have on the top, you will see the interpretation on this variant if someone else in the system has already identified and classified this variant. From this middle bar, you also have access to the alignment with the IGV browser. So here you have the different individuals that you have in your query. And if you click in the IGV position, you will go to the alignment of that specific variant. And in Solvar-D, we now have available data freeze 1, 2, and 3 in the system. So you should be able to query and see the alignments of the variants on all your patients there. We still have Exomiser to prioritize variant, and these functionalities, you can find them here. So you can use Exomiser in the system and also Genomiser, meaning that we also integrate the REM score to prioritize non-coding variants. And it's the same as previously, but you can also delete the HPOs that you think can give some noise, and then you get same as before the results of Exomiser directly in the system. As you know, sometimes it takes several seconds to work. So now when you click into the results, you have your Exomiser data in the system and the list of prioritized variant based on the HPOs. Apart from that, we have integrated another plugin for those of you that are familiar with Varsome that can give the clinical significance of the variants. We have collaborated with them to enable to add this information at the annotation level. So here we'll show you an example. If you have the Varsome API, you can paste here your password. For a demo, we only have information on chromosome 17, but so that you get an idea. And what we do is to annotate for the variants, the clinical significance and the ACMG criteria. As you can see here for this variant in chromosome 17, it tells you that according to the Varsome algorithm for the ACMG, from the ACMG guideline, this variant is likely benign, and these are the criteria that are supporting this information. Apart from this, we have added in the links that you can see on the left. So before you had to click on the gene at the variant level, now we have put all the information together. So once you select a variant of interest, you can go directly, for example, if you want to do the interpretation of your variant to the ACMG guideline, and you have a link to this position, to Varsome, Franklin, and winter bar, which will give you and apply the ACMG guidelines. For example, if I click here in for Franklin, I will get for that specific position that this variant is a variant of uncertain significance. We have also added other links that might be useful for you. I won't go through all of them. You can yourself go and see the different databases that we enable. I will just mention that we have included specific links for non-coding, to interpret non-coding variants like a trap score and regulation spotter that will give you information on of this specific variant. Trap is a score from 0 to 1, and then in regulation spotter they will give you information if it's affecting somehow a functional or a regulatory region and in which tissues it might be involved. Apart from this, we have also added links, for example, to gene reviews, and this is very useful once you have identified your causative variant to report on the recommendations that should be done to the patients. We have also added information on actionability, meaning that now you can see once you identify the causative variant if there is an available treatment in different databases. Tritabulum is the one that has been developed within Solvardi, and this is a work led by Giselle Bon in Paris. And then we also have the link to the EMBase or the ClinGen knowledge repositories. For example, if you click on the Tritabulum database, I think I have it here. So in this case, this gene is not there. I've tested, but what I want to show you is that if the gene is in the platform, you will directly have access to the treatments and information on those that are available in this database. And finally, we have also added information on pharmacogenomics. So we link to PharmGKB or at the variant level, so you can see for each of the gene if there is any pharmacogenomic application associated. What else? Yeah. So at the variant level, you can also now, apart from the tag that you are already familiar with in the GPAP, that by clicking here, you can tag the variant, and this is saved in the system and shared across all users. So you don't have to reinterpret a variant. You can also label the variant, and this is a private label, meaning that it helps me if I go back to this study. For example, if I want to follow up on that variant, I can click it here, or if I think it's an important variant, you can also select the other one. And the idea is that when you go back to the analysis after X time, you can easily see where you left and which variant you want to continue assessing. Once you save your study, you can also share it as before, copy pasting the link that you will have. And you will share here the whole study with all the information and the labels on the different variants for your specific group. So I think for the results section, this is everything we wanted to show. So now I will go for the cohort analysis. So if you can always go back to the main page by clicking here in the house on the left. And now I will select the cohort analysis. So once I select this type of analysis, here I will have a list of the cohorts I have created in the past. We won't do the demo of the cohort tab today because it will be a lot of time. But it's just from there, if you don't have any cohort created, by clicking to the cohort tab, you can easily create a cohort. For example, here I only have a few trios from this demo platform, but I could select the 15 index cases that are in the system. And remember that you can always also filter by the phenotypic data that has been entered in the system to try to define your cohort of interest. Apart from that, you can always download the information and create, I don't know, also a cohort outside the system and upload then your participant list so that you complete the cohort you would like to analyze. So once you define a cohort, you save it in the system, and then it will appear in the list of the analysis to do. So here I will just select, for example, the index cases that I have in the system, and try to make a query with this data. So you can select the genotype you would like to assess. You can assess heterozygous and homozygous also at once. And well, for the purpose of today, I will just select a predefined gene list. So you can do the analysis in one gene or a list of genes. And then again, I can define the filters by my predefined filters, or I can also add, in this case, I can add the variants that have a high or moderate impact in the genes. So when I run the query, this is, we try to improve from what was before, so it's easier to browse the information and identify the interesting data. So on the left, you have all the genes from the gene list. And on the right panel, you have the information on the participants and the corresponding variants. So if I open the participants here, I can see information on the HPO or the diagnosis that has been made for each of those that have variants in the system. But if I am interested in a specific gene, for example, I can select the gene on the left and then see the variants that I have associated. Here you can see the number of experiments that have a specific variant. And if you select a variant of interest, then automatically the upper panel with the participant will be updated with only the participants that have this specific variant. And the same the way around. If I select one of the participants here, I will only get, I can see all the variants from this participant, specifically clicking here, and then this will automatically give me for that specific participant the variants that are in the genes I have asked for. In case you identify a variant and a case that is of interest, you can click here in the contact button, and this, as before, will send an email to you and the owner of that case so that you can get in contact and start a collaboration or discussing the case to find the possible diagnosis. So this is for the cohort analysis. And now I would like to show you the matchmaker functionalities. So the search across all will be very similar, identical to the cohort analysis without having to select a cohort at the beginning. You will just do the same across all samples in the platform that I mentioned. It will be available in the next weeks. So for patient matchmaking, I will switch to the main platform so that I can show you an example. So here, first, the system will retrieve all the patients that have given consent to be discoverable through Matchmaker Exchange. And then I can select one of the patients. And here I will have the information retrieved from Phenostore with a list of HPOs and the gene, candidate gene that I would like to try to find another patient with this similar information. So now, for the purpose of the demo, I will just make a query within the database to see if there are similar patients with a similar phenotype and variants in that same gene. So now the system is checking all the participants, these 8,209 patients that have consent to be queried. And the result is the list of similar patients with a score that goes from 0 to 1, 1 being a perfect match between the phenotypic information and the gene. Again, if you see that there is an interesting case similar to yours, you can use the contact button that will do the same. Send an email to the submitter and to yourself, so you can get in contact and start a collaboration. So this is for Matchmaker Exchange, and here you have the different endpoints from where you can do this query. So now I've done it internally with all the patients in RD Connect, but you can also query Phenome Central, Decipher, Magento, or the broad database. If you don't know if or your patient does not appear in the search box, this means that the consent has not been granted within the platform, and you can always change this if you go to PhenoStore. So let me quickly show you. So if I load a study now, I go, for example, for this patient in PhenoStore. And if I edit the case, I can go to the patient consent and give the possibility to be discoverable by a Matchmaker Exchange changing this functionality. And then if I update it, now the case will be discoverable in the system. And so finally, if I go back to the landing page, the last thing I wanted to show is the clinical report. So once you have analyzed your case, tag the variants in the system, you can access the clinical report module. And then if you select one of the cases, so we had case 15C. Then the system will automatically retrieve the information we have and the variants that have been tagged in the system, and I can select which ones I would like to have in the report. And here I have two options. Either I download the clinical report draft, where we have pre-filled, we have a template and we have pre-filled the information on the variant, where you will have everything that has been included in the system, and we are working also to have the different quality metrics there, but you will already have information on the pipeline, information on the clinical data that is in the system, and everything pre-filled so that it helps you to not lose time on creating that. But in case you prefer just to download the variant information, you can also select the download variant, and then you will get a CSV file with all the relevant information in a table format with the interpretation and several fields on information related to the interpretation and the possibility to do the interpretation with the ACMG criteria. So I think more or less I've shown all the new functionalities we have shown in the system. Yeah, so I don't know now if there are questions in the chat, or there are specific things that you could not find and you want us to look at it to understand where is the information now, because we have changed quite a lot how you see the data. So Steve, all yours to...
Thanks a lot, Leslie. So we managed to deal with most of the issues in the chat. I don't know if anybody's got any more questions now. So Mattis had one, but I think Alberto's just addressed it about the cohorts. or who are the cohorts visible to? And Alberto has responded that they're visible to the creator and the users belong to the same group, a GPAP, and that we're working to extend the permissions as done for studies in the new interface. If anybody else has any other questions, now would be a great time to ask. Don't be shy. So Anna's asking, and the top is asking, are the the Matchmaker Exchange functions done as a query, or is it a live search when you send a Matchmaker Exchange?
Not sure what you mean by that, Anna. So it's a live search in the sense that it's at that specific moment with the HPO you have entered and querying the other databases. I'm not sure I'm understanding the question. I don't know, Steve, if you can, or Anna, if you can explain a little bit more.
So in theory, it is a live search, I guess.
Yeah, in that specific moment with the HPOs that we have and the HPOs that are in the other databases. So it's live in the sense in that specific moment with the data that is available in the system when you launch the query. I don't know if that was the specific question.
Yeah, so essentially if anybody adds a sample to another endpoint tomorrow and you do the search again tomorrow and that sample is one of the matches, you will get a different result. You will get that match, you know. So it's live in the sense that there's nothing stored. As soon as things are made available in any endpoint, if you rerun the query, you will get a different set of results if there are other things that match, obviously. Anna's happy. Andres Caballero has asked, is it possible to add the gene list that is imported from PanelApp or the gene list that is given from the HPO terms? So after you retrieve that gene list, can you then maybe drop a gene out or can you add a gene in?
You cannot drop a gene, but you can add additional genes. For example, let me enter a case. So similar as what you had in the platform. These I haven't shown also, but so here I have my results. If I want to change something in my filtering, I can click here on new query, and then here you can clear the gene list if you have any, or add any specific gene of interest if you want to add it on top of the information you already had, and then launch again the query based on this information. So I could rerun the query and then well, have variants. In this case, they are not in this gene, but you would have more variants or less depending on the additional filter. What I haven't mentioned and I'm seeing now is that for each of the queries, you can here at the top also give a name, let's say, so that you remember. I don't know, here I put the DMD gene, so I can name this query as DMD. So quickly, when I go back, I can remember that, okay, this is autosomal recessive homozygous variant, and I was looking for these genes. And you can always see the filters that have been applied by clicking here in the view applied filters, where you have a summary of the information and the filters that you have applied in that specific query.
Great, thank you, Leslie. And I'll just add another one before coming to Heba's question, just for maybe for new users as well. Can we still upload a user-defined gene list?
Yes, so I go back to the new query here, and if you go by filter by genes, I haven't entered, we have not removed any of the previous functionalities, but I have not shown them again. So here in the gene search and upload, you can either look for a specific gene as I've done, or you can upload any comma-separated gene list that you would like to assess. And we also have, as previously, the predefined gene list, that the list that we have had in collaboration, for example, within Solvardi for the different European reference networks. You also have this gene list completely available in the system if you want to filter by those on your data.
Okay, So we have another question regarding matchmaking from Heba. So Heba is asking, once the cohort analysis is fully possible, if you have a patient with a candidate gene from local data, can you do matchmaking in SolveRD?
If you select if the patient is in the database, yes, you just need to select your patient from the drop down, and then you can add any gene. Let me go back to the main platform. How can I show this? Let me go back here. Here I could add any gene. Imagine that yeah, the same. The DMD gene is the one that it's my candidate gene. I can add it here, launch the query, and then when you go to the other databases, you can see if there is another patient there similar to yours with a variant in that gene, if it has been tagged, or depending on how they access this query in the different systems. If you want them within SolveRD, you will need to select, as I've done here, the RD Connect database. But the thing is that we need to have this gene tagged in the system, so that other group should have also identified this candidate gene, so that you have a match. If you want to go randomly across all the database, then you will need to create your cohort based on the phenotypic data, and then look at the variants and identify yourself this second patient. So these are two different approaches.
Yeah, I think the second one was probably what you were thinking of, Heba, at a guess, where you have a candidate gene and you want to see whether there's similar patients in the database. So that's exactly what Leslie was saying. You would build your cohort and add your gene and see if you get something that makes sense as a sort of match to your candidate gene, if you don't have it, if you don't have the experiment in the system. Manuel Posada, is the creation of cohorts limited to probands, or can we also include controls such as their unaffected parents?
Yes, you can. If I go back to the cohort application, okay, let me create a cohort. You will see that, well, this is the demo instance, so we have very few information, but you can create a cohort based on the experiment information, meaning, for example, if you want only exomes, only genomes, etc., or also the affected status. For example, if you would like a cohort of unaffected only, you can select those, only consanguineous, males or females, cases that are unsolved. In principle, we have, I would say, almost all the information that you have entered in PhenoStore should be here queryable, so that you can create your cohort. And we have separated this in the information from the patient. That would be this first tab, and then the specific clinical exploration would be with the HPOs or the ORFAT terms, so that you can select the cohort based on the clinical data. And then once you have the cohort created, here in this data table, you can also delete some of the patients if you don't want a specific individual to be in your cohort. So it's quite flexible in this sense. From here, you can also only select your samples. You can select the samples from a whole project if you want only, for example, to analyze SolveRD samples, or to the whole RD Connect cohort. So it's quite flexible in this sense. We have tried really to be to have all the use cases possible based on the information that you have already entered in the system. So it's useful for you to have it there.
And Anna is also asking, when we're matchmaking by gene, can you specify filters for those searches, like minor allele frequencies or the type of variant?
No. So if you are using the Matchmaker Exchange functionality, this is really, for now, a query which is based on at the gene level. So only if someone has tagged a variant in that specific gene, and the HPOs for the similarity on the phenotypes. If you go through the cohort or the search across all, as we were, you're also able in the database, then yes, then there you can add any filter in the cohort analysis. I will go quickly a cohort so you can see, because maybe I went fast there. You can select any gene of interest. Here I select the gene list, and then here you can apply any filter, meaning that you can look for red variants, only the one that have a high impact, for example, that can be truncating, etc. So you can restrict your search for the search within RDConnect. They are working in Beacon version 2 and trying to have improvement on the API so that you can do this query also across other databases, but this is not something feasible today. But it's they are getting there, and I guess in the next month it might be an option so that you can yourself look for a specific type of variant in a database without having to tag a specific gene at the patient level.
Manuel Posada very kindly says this new framework is impressive. Congratulations to all of you involved in the new development. Have we elaborated some written guide or manual?
Yes, so we have to improve it because we are missing some step. But once you access the platform, I will go back now to the main page. But you always have here on the top right the user guide. And this will bring you to the different functionalities or approaches for the genomic analysis, but also you have guidelines on the experiment data for the data submission and data management, and also in PhenoStore for the phenotypic data, how to submit, how to manage. So here you have more or less all the information, and we are still working on finalizing the missing bits, but I think most of the functionalities are described here.
And Anna Esteves asked us, does the system include triplet expansions, translocations, or other types of variants apart from SNPs, indels, and CMVs?
Not yet, but we are working hard on having these. For example, structural variant, I think STRs will be the next type of variants that will be available in the system. But we need to implement them at the pipeline level and then for visualization. But this is on our to-do list and work plan.
Yes, as Leslie says, we're taking it bit by bit and getting there, but we're certainly close to being able to, through our work with ORDO, obviously we're more familiar with these types of variants now, and we hope to be able to incorporate them soon in the platform view. Kieran has a question. Can we save our custom variant filters apart from the preloaded ones? And I would also ask, can we share our custom variant filters?
You cannot share them, but you can save them. I will show you how. So now I'm loading the study we were working on. And so here, when you go to new query, so when you do a selection of filters, this one, for example, then you can save this filter set. I enter a name. And once it's saved, then it will appear in your list of preset filterings. Yeah, it's not shareable yet, but it's something that we can think about, so that you can all use the same predefined filters.
And yeah, this is also something we could maybe add as well. We could have a set of predefined more. We could add extra predefined filters, I guess, ourselves, right?
Yeah.
Okay. that seems to be all the questions for now, unless I've missed anyone. Okay, I don't see any movement in the chat. Quite a few people are saying thank you very much for the presentation, Leslie, and well done the team for the developments. People seem to like the new interface. We hope you'll all get used to it quickly. Obviously, for those of you who've been using it a long time, it's a big change, and some of us aren't so good at changes as or at dealing with change as other people. But obviously, as usual, if you see anything that seems to be amiss, or you spot any bugs, or you have any problems, just get in contact with us. Usually through the help desk is the first place. And obviously we'll resolve them or explain them if they're not actually bugs, but they are features. So we have a couple of final amazing work. Thanks for the updates and the presentation. So just to say thank you to everybody for joining the call today. We appreciate your time, and bon appetit, and we'll see you again soon.